little shorter here. Can everyone hear me? Perfect. Hi, I'm Laura Schaevitz, SVP, Head of Research here at Recursion. A little bit on my background. I'm actually a PhD neuroscience from Stanford University. And before coming to Recursion, I was actually the Chief Scientific Officer and then CEO of a company called Viome, which built the technology underlying the infovomics that Lena and Imran just told you about. And so one of the things I just kind of want to start with here is when Recursion offered to acquire Viome 2.5 years ago now, I think it was, I was really excited. So the technology we were building at the time, we were trying to get adopted into the mid and large pharma companies, and adoption was maybe a little bit slower than we would have liked. And I think a challenge there is just finding companies with the right mindset. And Recursion is that place. I think looking at what Viome was trying to build and what Recursion is trying to build, there was just a beautiful mesh. And so what I'm really excited about sharing at the end of the presentation here is how we're actually employing that technology on our programs today. But before we get there, I'm looking forward to sharing with you how we're using the Recursion OS to both discover and drive some of our internal programs. So to start, I'm going to give you a little different view of our maps of biology than what you saw before. So this is the heat map version. And just to give you just a little bit of an orientation, what you see on the, let me see if I get this right, what you see on the rows and the columns here are the individual genes, and we're looking at relationships across these maps. And so along that diagonal, you see self-similars, they look great. What you also see are these clusters of biology, where what we're seeing are those genetic perturbations that look very similar to one another. By taking this really, and what I guess what I would also say is this is just such a tiny snippet, as both Chris and Imran and others mentioned. Our actual maps contain the whole genome knocked out and hundreds of thousands of compounds. And by taking such an expansive approach to how we look at biology, we can really use these maps in very different ways to uncover kind of those novel insights in programs. And I'll walk through a couple of programs today with you. But just to give you like an overall sense of what that might look like, we can use our maps to uncover what was potentially considered at one point an undruggable target. So for example, if we put that target into our map of biology, we can look for other genes that look phenosimilar to it, which would give you a sense for maybe these genes act upstream, downstream, or even potentially adjacent, but still provide that same phenotypic output. The other areas that we can potentially target, see if I get this right, are adding in some of our patient genomics, the large patient genomics data sets that are out in the world. We can combine those with our maps to really uncover those interesting novel insights. So let's actually dig into a couple of our programs. So the first program I'm going to tell you about today is Target Alpha. And for Target Alpha, we're combining these external data sets with our maps to find what we think is a potential first-in-class NCE with novel, we say MOA here, but the reality is this is a compound with polypharmacology where both of the activities of the compound we think are really essential to be driving this enhancement to immunotherapy. So we all know the story of immunotherapy. We all have probably heard 1,000 pitches for immunotherapy drugs or combinations with immunotherapy, and I'm going to tell you that this one is a little bit different. So in traditional immunotherapy, we know that of the 44% of patients that are eligible, a huge fraction of those patients don't actually respond to immunotherapy alone, upwards of 88%. We know that of those who could potentially respond, a large percentage of those patients have these immune-related adverse events which prevent them from actually being able to take the drug that could be effective for them. And so at Recursion, we're ambitious, and our goal was not just to find another immunotherapy or another drug that could be used in combination with immunotherapy, but to be able to find a drug that could both increase the number of patients that would respond to immunotherapy drugs, and also minimize those peripheral side effects. And I think we might have something. So let me tell you how we used our maps to find the starting point for this program. So our oncology team took a couple of very famous papers, Manguso and Lawson, which describe about, I think, a couple thousand causal markers of either enhanced or resistance to checkpoint immunotherapy. They pored over them. They narrowed it down to about 110 genes that they were particularly interested in for a number of reasons, including perhaps which ones show nice phenotypes in our maps. They mapped them, and then they looked for interesting relationships. And so let's actually walk through the map. So here is the actual map of all 110 genes. You see the nice self-similars, and you also can see a lot of very nice clusters of these genes. And one of the clusters that we were most excited about is a cluster that is right up in here that contains a gene called BRCA2. And if we zoom in on that cluster, you can nicely see BRCA2. You can see BRCA2 with a lot of other BRCA2 family members, gives us great confidence. And then there's this extra gene called gene A, and I won't tell you what that is. But gene A is a known immune modulator. So that was kind of interesting. So we took that gene and we put it back in those broad maps. So all genetic perturbations, all of the chemistry we had, or all of the compounds we had at our disposal. And we found two interesting relationships from that. The first interesting relationship was the gene A we found an actual inhibitor to gene A, which is always good when you can find that. But we also found a second gene, gene B, that gene A and the inhibitor to gene A seem to be phenosimilar to. And this one was really exciting because gene B turned out to be an up-and-coming resistance marker for immunotherapy. So now we have potentially a compound that is phenosimilar both to gene A, which is immune modulator, and gene B, which is a resistance marker. And we actually went ahead and we confirmed that this compound was active against both of those targets. So now we're getting pretty excited about this program. We potentially have a compound with polypharmacology that can be hitting kind of multiple different levels of this program. So we went ahead and put it into an animal model. And for this program, we used a CT26 animal model. And we use that because it's fairly highly resistant to immunotherapy. And as you expect, I'm a pointer, so as you expect, here for the vehicle and the anti-PD-1, you see really no effect on tumor volume. But now we're starting to get excited because we see a monotherapy effect for our compound with an actual improvement when given in combination with anti-PD-1. Upwards of, actually for this combination, we saw upwards of 40% complete responses. When we took those 40% complete responders, we re-challenged them. We saw that 3 of the 4 had no tumor regrowth, suggesting immunological memory. So this is looking pretty good. We then looked within the actual tumor microenvironment to see what was potentially going on there. We saw an increase in immune infiltrates, as evidenced by these total CD45 positive in the combination. We also saw decreases in suppressive macrophages, as well as LAG3 exhausted T cells. So generally looking very good within the tumor microenvironment. We also looked at cytokine levels, and I'm showing you here just 2 examples of course, a panel of cytokines that we looked at. Within the tumor, evidence of immune response within, or I would say similar immune response across all of our treatments. But remember, our goal was not just to improve response in patients in combination with immunotherapy was also to decrease that peripheral inflammation. And so we got really excited when we looked at cytokine levels in the periphery. And so here you can see plasma levels of these same cytokines with the anti-PD-1 elevated levels, which are significantly reduced in the combination. So now we potentially have a compound which is showing efficacy in our animal model in combination, and when we give our drug, we're reducing this peripheral systemic inflammation that's potentially limiting the ability of patients to be able to take these, to be able to respond to immune checkpoint therapy. But of course, the compound is not ready to go into the clinic. There's work to be done. We need to optimize it against both of our targets, and we needed to do some work on drug exposure. So our amazing medicinal chemistry team actually turned to the Recursion OS in order to help them drive chemistry optimization. And now you're going to get to the geeky part of me, because I love where the technology and the biology actually intersect with one another. So for this program, we had all of our compounds synthesized in an external CRO. At the CRO, we knew we were trying to optimize against 2 different targets. So we set up, biochemical assays there, as well as reporter assays. But then we also had the compound shipped to Recursion, and we immediately profiled them on the map. And what we wanted to see above me here is the compounds all looked similar to one another, and that they looked, that their phenotype looked similar to that target phenotype that we really cared about, which should represent the polypharmacology that we cared about for this program. Now, the map helped us stay really laser focused on this target phenotype that we cared about, and I'll show you an example of how it did that. About halfway through the program, our chemists introduced a second core, and you can actually see here that in this new core, our reporter assays started reporting out that potentially we had increased cell-based assay activity that was potentially really exciting for us. Now, possibly in some other place, we would have been excited about this, this new core, but at Recursion, we never got excited about this core. I think the excitement lasted for all of maybe 5 days, because as soon as it was profiled on the map, if you look above me, you can see these 2 new compounds here, but their phenotype was not at all like the target phenotype that we cared about. And so we knew that we had done something in introducing this new core that was not something that we cared about. And of course, we're interested in excited chemists and biologists, so we wanted to understand what it was exactly that we had done. And this is like the true power of our map. We didn't have to run an experiment. All we did was we took these 2 new compounds, we searched them in our broad maps of biology and chemistry, and we said, is there a gene knockout that looks somewhat similar to this? And lo and behold, there was. So here we are. And that gene was CDK9. We actually went and confirmed in a simple biochemical assay that those compounds were active and inhibited CDK9. So a beautiful example of how we can not just drive the chemistry, but if something goes off the rails, we can understand what it is that we did without having to assess or set up a whole bunch of new biochemical assays to be able to do it. So let me just give you the kind of overview of target alpha, and then I'll move on to gamma. So I showed you here how we used our maps to combine external data sets with our internal map in order to find this magical compound that has polypharmacology that can potentially both increase response to checkpoint inhibition as well as to decrease peripheral inflammation, and then how we used our maps to actually augment our chemists to drive chemical optimization for this program, including when we went off the rails, what exactly had happened. So now let me turn to target gamma. And in this transition, I want you to remember, and I think this plays back to a lot of what Lena and Imran were talking about, around how we try to use exactly the same strategies over and over again to industrialize these processes. I'm kind of going to tell you exactly the same story for gamma, so please don't go to sleep on me. But I'll try to make it a little bit more brief this time around. So first, we introduced target gamma 15 months ago, and I'm actually excited as we're doing this to announce to you what the actual target for gamma is. And target gamma, we're going after an RBM39 degrader for this program. And let me give you just kind of a high level of the philosophy of the program, and then I'll get into how we used our maps to actually find RBM39 as this potential target for gamma. So the philosophy for the gamma program was the following. We were looking for a molecule to be given in combination with PARP inhibition for patients who are homologous recombination proficient, which is a large patient population that currently has no viable treatment options. So for ovarian cancer, first-line treatment, as many of you already know, is debulking platinum-based chemotherapy. Many patients respond very well to that and see complete tumor regression. If you give a PARP inhibitor, they're a large portion of the patient population, 50% that are homologous recombination deficient will actually respond very well to that PARP inhibitor, and you'll see an enhancement in progression-free survival for those patient population. But if you look at the 50% of the patient population that are HR proficient, you see almost no increase in progression-free survival for that population. And so this is the population of patients we were really going after for this program. So now, hopefully, the part you have all been waiting for, how did we identify RBM39 as this alternative target for CDK12? So we started with CDK12 because it's been known to be a target to improve response in the HR proficient setting. But we also know that it is a challenging target to go after because it is highly homologous to CDK13, which has a number of toxic side effects. And so our goal here was to be able to find a target which was adjacent to CDK12. We don't know exactly how RBM39, is it upstream, downstream, is it a side, or is it adjacent with that same final phenotype? But that's what we were looking for this program. When we turn to the map, getting again to a bit of what Lena and Imran were talking about around biology may not actually always look like what you expect. We actually found that CDK12 and CDK13 are not at all like one another in the map, and so that was kind of an interesting finding on its own. Then when you look, when you put CDK within that kind of broad expanse of our map, we found that CDK12 was very similar to RBM39, which is not known or documented at all in the literature, and that we also found this compound REC65029, which is highly phenosimilar to both of those. We did confirm that our REC65029 is in fact acts on RBM39 and does not act at all on CDK12 or CDK13, so it is very specific to RBM39. In this program, I'm not actually going to show you at all how we do it in the interest of time, but I will tell you that unlike target alpha, where we set up biochemical and reporter assays, we actually drove SAR for this program, or chemical optimization for this program, 100% using our phenomics maps. That was the only tool or platform that we used in this process, which I think is really amazing and impressive. I totally skipped ahead because I was so excited about that part. My apologies. Let me first get to the important piece, which you probably all care about, which is the animal data. One of those little hiccups, right? So for this program, we put this molecule in combination with PARP inhibition into an HR proficient ovarian cancer model. As you expect in this model, PARP inhibition alone has no effect. Please notice that that is a log2 tumor volume scale. We were really excited to see that our compound alone showed very significant effects. And when you give it in combination with the PARP inhibitor, aliparab, you can see even increased efficacy in that model. If you look over here, on this side, you can actually see very significant or faster rate to complete responses in the combination versus the single agent alone, and a durability of the response in the combination versus the single agent alone. But it is exciting that we saw single agent efficacy for this model, or for this molecule as well. So now to the piece that I was excited about, the fact that we were able to really use the platform to drive SAR alone, I think is really significant for us at Recursion. But now I want to take you, so we did some work, we optimized the molecule. In this case, we were looking for a molecule that had a better therapeutic index than this starting molecule. And let me take you to the piece that I'm potentially really excited about because I'm biased. And this was the work that I did for 5 years. And that is how we used the in vivomics platform to help us pick and figure out what molecules and at what doses we should take into efficacy studies and potentially into the clinic. So again, here is the beautiful images of the animals in the cages. And as Lena was referring to earlier, we take 24/7 video and we automatically read out behavior from the animals. And what I'm showing you up in this corner here is that first compound that we identified, Rec 65029, and the actual profile for that. This is activity measurements. And as many of you know, animals are nocturnal, so you see these beautiful circadian rhythms with high levels of activity at night, low levels of activity during the day. For this molecule, in black are our vehicle-treated animals. You see some depression of activity in some of the higher doses, really speaking to the need to get to potentially a better therapeutic index than what this molecule could provide. As we ran through our chemistry optimization, we identified a number of molecules that were potentially quite interesting in terms of having the drug exposure properties that we were looking for, the potency that we were looking for. And here are just 2 examples, and I think this is a really good indication of how we can use technology like this to help us move faster. So the first molecule I'll draw your attention to is this Rec 11700204, in which you can see, again, vehicle looks fairly normal, but at both doses that we gave, which were required for efficacy, we saw very significant decreases in the activity levels of these animals, suggesting potentially some safety concerns for us. Whereas in this molecule, Rec 1321245, you can see little or no changes in the activity levels at all for these animals at doses that were required for efficacy. And so this really is an example of how we can quickly assess the potential safety of a molecule in an exploratory study. And this really helped us hone in on this is probably the more kind of safer, efficacious compound to move forward. And what I would say for all of these is that we have filed a number of patents around this, both around method of use as well as a number of these molecules in terms of composition of matter. So let me kind of end with just a reiteration for the target gamma program. While I didn't show it, well, I showed you how we identified kind of that the relationship between the novel relationship between CDK12 and RBM39 using our maps. I told you a bit, but I didn't show you how we used our maps and our phenomics to really drive SAR for this program, and then how we're starting to incorporate some of these other high throughput assays that we're working on to help us figure out what are the right molecules that we need to be driving forward as quickly as possible into the clinic. So with that, let me give you the quick where are we going in 2023 for our preclinical programs. We are working to drive target alpha and gamma to IND-enabling studies in 2023. We're continuing to augment and bring into our internal programs digital chemistry and predictive capabilities, which Imran started to touch upon, but I wasn't able to show you any really good examples here today, but hopefully the next time we will. And then the areas that I'm really excited about over the last couple of years is we've seen how we can use our platform to really drive these programs in highly consistent ways. Gamma is a perfect example. We're no longer setting up bespoke assays to be able to identify and drive our programs. And so really in 2023, we are doubling down, and I love being partners with everyone here, including Lena, on really setting up these industrialized workflows for our preclinical programs, such that as much as possible, we're taking the scientists, I wouldn't say totally out of the loop, but we're taking them a bit out of the loop. It's sometimes uncomfortable for them, but we're taking them out of the loop on some of these early stages of inference and hit to lead, so we can drive programs faster and at scale to really deliver on the mission of what we have here at Recursion, which is to decode biology to radically improve lives. And with that, I think, thank you, and turn it over to Tina.